In this video, we will talk about the basics of cell culture and aseptic technique. We start off by showing you the very first culture that was successful, that scientists were successful in culturing in vitro, which means outside the body, away from the native setting in a lab setting, and those were the Hela cells. Hela cells were isolated from Henrietta Lacks, and you watched a very profound documentary on it for your discussion assignment. These cultures have been growing for obviously decades at this point and continue to grow and be used in molecular and cellular biology research. We usually use them to grow proteins that we are interested in studying further. So we kind of use them as a factory for proteins and genes of interest that we are studying in our cell culture setting, because they grow extremely fast and they literally overtake within a couple of days. We usually don't use them to study biological processes as much anymore, as they change very quickly and they become more and more transformed over time. So it's hard to get reproducible results from this particular cell line. However, these cells were instrumental in us developing the field of molecular and cellular biology as we know it. Without the help of these cultures in developing the proper protocols and procedures needed to culture cells in vitro, we would not have been able to study them the same way, or it would have taken a lot longer to develop these. So how do we actually create cell lines? Well, think about a small piece of tissue that you would get from a particular organism, whether it be human or mouse or another model system that you're working with. Well, that little tiny piece of tissue, usually we'll get it as a little chunk from surgical tissue or during surgery, or we may be getting a biopsy, so very small piece. And now we want to culture it. Well, that little piece of tissue, let's say that it was a liver tissue, will contain a host of different cell types. Obviously, it will contain hepatocytes or liver cells, but then it's going to contain helper cells like Kupffer cells. It will contain all the cells associated with the blood vessels, right? The cells that make up the blood vessels, the endothelial cells. It will contain the blood cells inside those blood vessels, so the various types of blood cells, red blood cells, all the different types of white blood cells, platelets. So it will have a host of cells associated with that. Then it may have lymphatic vessels going through it, so that will have its own cell types and its own types of cells inside them. It may have nerve endings or nerve cells associated with that particular part of the tissue. You may also have other types of cells in there, right? And all these cells are in there in a very organized manner. They're not just kind of haphazardly put together. They're put together in a very organized way because of the presence of a basement membrane and the extracellular matrix that's part of that tissue to keep it the way it is, providing the scaffolding for all the different types of cells present there. You'll also have fibroblasts and all these other proteins and cells there as well. So in a very tiny piece of tissue, you may have, 20, 25, even more types of cells in that little piece. But you would only want to have a particular type isolated from there to culture in order to study it further. So the first thing we usually do is kind of melt away the basement membrane or that scaffolding by using specific enzymes that can chew away the membrane proteins. One of the protein that is very common in these scaffolding is collagen. So we can use collagenase to kind of melt away the tissue and create a slurry of cells. Those cells are then taken through a series of purification steps, usually involving gradient centrifugation that will isolate them or separate them by different size and weights, and then we can get the portion that we care about. Well, in our example of liver cells or hepatic tissue, we're lucky because hepatocytes are relatively large cells and easy to isolate because of that. So we can do just a couple of centrifugation steps and get the large cells out into a culture setting. That very first step is the time that you're going to culture these cells. That particular first culture is called a primary culture. These cells are as close as you can get to the way they behave inside our body. They will look the same, they will act the same, and you can study them to look at the biological processes of interest and learn about them as they happen inside the body. They'll work very similarly. However, it's not going to be the most clean culture, since you will get some straggler fibroblast and other types of cells carry over into this primary culture. As we grow them and provide them with the right environment specific for hepatocytes, we will start to clean it up. So the most purified culture many times can be your secondary culture. So the first time that you subculture these cells after having grown them into your vessel to point of confluency, or when they're fully taking over that space, and you move them to a new home, that can be a very clean culture, much more purified culture. So that would be typically called secondary culture. The way we do that is by removing the cells from their first home, so to speak, which is usually either a cover slip or a plate or a flask that I will show you in a few minutes. And then you remove the cells from that and you move them to new fresh vessels. And that process is called passaging or subculturing. And the first time that you culture these cells, it is called passage 1. So if you were to look at these cells, the primary culture is passage 0 because it's never been cultured outside the cell, outside the body. And so that would be designated as passage 0. And then when you subculture it, the secondary culture, the very first time that you passage them outside the normal body setting, in vitro, it will be called passage 1. Now you can continue to do this process multiple times, and each time the passage will become different, right? So the next time it's going to be passage 2, and then passage 3, and then passage 4, and so on and so forth. Now, if you have normal cells or typical, completely normal cells that have no issues with them, you can culture them primary cells. You can subculture them about 8 to 10 times before they reach senescence, and they don't grow anymore. The reason for that is that the telomeres are shortening each time they're replicating, just like they would inside our body. And over time, those 8 to 10 cultures equate to about between 40 and 60 replications. They're getting to that stage where they would be what you would call old cells. So same as our body ages over time and telomere shortening is part of that story, same way these cells also are aging over time as they're replicating and dividing multiple times, and they reach senescence and do not grow. They first slow down their growth and eventually don't grow well at all, or don't grow and replicate at all, and eventually will die out. If we want to keep a cell line for further studies, we can do one of two things. If you want to keep them in a primary culture setting, we take our cells at the secondary cell culture stage, so passage 2 or maximum passage 3, and we freeze a bunch of them at that time so that we can always fall back on them when we need them for experimentation. So each time we take a little aliquot of them out from the freezer and then thaw it out and reculture it and use them for our experiments. And we usually don't go beyond passage 8 in those cases because they will start to look different. The other way we can do this is through a process called immortalization. Now, this is important when we want to keep a particular type of cells for long-term experimentation, and we want to continue culturing them for a longer period of time. So in that case, there are two ways that can be done. One of them is through spontaneous transformation. Cells by themselves, when they're getting cultured, the subpopulation of them will get mutations in them that make them transformed and immortalized. These cells will continue to grow and divide and replicate even beyond that passage 10 stage and will not reach senescence. And so that little subpopulation of cells obviously is not the same as your original completely normal cells. They do have a mutation, and it's somewhere in one of the important pathways associated with cell cycle control or cell cycle itself, so that they can continue to divide and replicate. However, they are now immortalized cells and can be made into a cell line that can then be used for further research and continued research. In this case, you probably don't want to use it to study cell cycle because you don't know where the breakpoint is. So you do take the data with a grain of salt. However, you can still study them for all the other functions and biological pathways associated with those particular type of cells. Another way that you can immortalize them is by doing an actual transformation yourself, in which case you create the mutation using genetic engineering in a particular pathway in cell cycle. Now, those cells can still be used in cell cycle research since you know exactly where the mutation is, and you can judiciously use them for particular types of cell cycle research regardless of that immortalization. However, by doing the immortalization, now you're able to use these cells for many more replications, usually even 40, 50, 60 passages before they become so different that you want to throw them away. Now, cells over time, as you culture them, will get more mutations, and cells that are immortalized are more prone to mutations because they're already a little bit disrupted. They already have a mutation in them. And so eventually they will become so different that they are no longer that original cell line that you created or that you generated or started to work with, and they will need to be discarded. However, this allows you to keep them for a much longer time and be able to use them for a much longer time. Another type of cell line that you can generate is through single cell isolation. This becomes extremely important in cancer research, where many times a single tumor may have cells with various types of mutations. So you can take a single cell and create a cell line out of it, and that cell line will be a clonal cell line because every single cell in that cell line will be coming from that one original cell. Now, depending upon what type of cells you're culturing, they will look very different under the microscope. So you can actually identify the type of cells that you're growing based on their morphology that you look at. Some cells are fibroblastic, so they grow in, they're these thin, long cells. They grow in a very organized manner. They almost make this like Fibonacci culture, or pattern within the culture when they're fully covering the plate or the vessel that they are being grown in. But they grow beautifully and they look very pretty. So that's one of my favorite cultures. So this is an example of a fibroblastic culture. This one obviously is very few cells are present in that area, so they're still not showing the beautiful patterns they show later on. Another example is this epithelial culture, where you see the cells are nice and flat. You can see the nuclei inside, and then you see the cell body, the actual cell membranes, cell boundaries of these cells. They will look, they sometimes have a glossy look because of the membrane proteins that they are secreting in order to create the junctions between cells and between individual cells and the cell and the matrix. Here, another example is of a culture of endothelial cells. They look similar to epithelial cells, but they are more flat and clean, so-called. They don't have the same kind of secreted proteins or that you would see in epithelial cultures. And so these are the cells that line all your blood vessels, for example. Again, you will see nice clean membranes, and you will see nuclei inside, very visible within these flat cells. And then here is an example of a neuronal culture where you have cell bodies along with their processes, similar to how you would have the neurons inside our brain with their axons and dendrites. So these are similar processes that are happening in this neuronal culture. as they're differentiating into neurons. That is another type of culture we look at. Now, when we're looking at cultures, we look for several things. The first thing, anytime you're given a plate of culture and you look at it under the microscope, there are a few things that you need to be mindful of. The first thing you will look at is obviously the morphology, to make sure they are the type of cells that you expected to get for that particular day or for that experiment. And that obviously simply look at the cells and make sure they look the way they should. The second thing you want to see is the health of the cells. So the cells should be, majority of them should be nice and flat, attached to the surface, to the bottom surface of the plate. They should not be floating. You'll always have a few floaters, and those floaters indicate dead or dying cells or stress cells. But the majority of the cells should actually be attached to the surface. Here you see, the floaters can show up as these bright dots in your actual sample right here. They're rounded cells right here. And so those indicate floaters. You shouldn't have a lot of them, but you will have some of them in every culture. Then the third thing that you look at is how confluent or how well the culture is growing, how confluent your culture is. Now, a cell culture's confluency basically means the percentage of your plate or the vessel that is covered in cells. So looking at this particular area, you see a lot of empty space and very few cells. So this would be about 15 to 25% confluent culture. Looking here, this one is more covered. About 40% of this particular surface area is covered. So this would be a higher confluency culture. Here you have very few spaces that are empty. Majority of the area is covered in cells. This would be a 90% or more confluent culture. In a neuronal setting, you would want to look for the cell bodies themselves in the confluency measurement, and so you don't actually take your processes into account. So here it would be something like a 75 to 80% confluent culture. For confluency measurement, we always want to look at the cell in a little bit, the culture at a lower magnification rather than a higher magnification, because you will get a more, a bird's eye view rather than a very small area of your culture. Because different areas in your culture plate can have different confluency. They may not be growing all homogeneously all over. Okay, so what do we need for these cells to grow? Well, there are multiple things that we need to grow. And so you can think about it just like any living organism. To live, every single organism requires space. They require nutrients, they require the right environment, and they require protection from infections and viruses and bacteria, obviously. And then finally, they need to be able to replicate and divide in order to produce more of themselves. Similarly, when we look at these cells outside the body, outside an organism, in a culture setting, they need the same things. So the first thing is they need some kind of substrate that they can attach to. Majority of the cells in our body cannot be grown in suspension. A few outliers would be obviously your blood cells that would be growing in suspension, or cells associated with the lymphatic fluid or the cerebrospinal fluid that may grow in suspension. But majority of our body cells require a substrate to attach to. And that is provided to them inside our body by the presence of the extracellular matrix and the basement membrane. Outside the body, in a culture setting, this is provided by chemically modified plastics or coated materials that are coated with either ECM proteins themselves or ECM-like material itself. And that provides them with the scaffolding material to grow on. The second thing is nutrients, and that those nutrients include obviously, water is basic requirement, but in addition to that, they need the sugars, they need the salt balance, they need the the same things as we need. We need carbohydrates, and we need vitamins and minerals, and we need our fats. Similarly, these culture cells need the same things. So they still need the sugars, they still need the fats, they still need all those same amino acids to make more proteins, all the same things as what you need. and those will be provided by specific components in the culture media. And we'll talk about those in a second in detail. The third thing is their environment. That is, they need the right temperature, humidity, oxygen, CO2 control, and this is provided to them by the presence of our special cell culture incubators that are water jacketed and attached to a CO2 tank with humidity and temperature control. And finally, they need appropriate sterile environment, and we provide them this by using aseptic technique for culturing them, and then by adding antibiotics and antimycotics into our culture media so they do not get infected easily. In addition, cell lines are routinely tested for mycoplasma to make sure that they are not getting infected with microbes as well. Now, for their culture media, we have the Basal Media that we are starting with. This is essentially what you would call Gatorade for cells. That's what we call it, in layman term. So, because Gatorade, think about what it will contain. It contains water, it contains salt, and it contains sugars. Those are the three main components of the basal media itself. The one that we use most often in our lab is called the glucose modified Eagles medium, or DMEM. In addition to the basic Gatorade stuff, right, the salts and the sugars and the water, it contains appropriate buffers to maintain the pH and osmolarity of the solution. It also contains a pH indicator in the form of phenol red. So the red color is coming from phenol red. You can also find it without the phenol red if you need just clear media for some experiments. And this phenol red would change color if the optimum cell growth pH is not maintained, which is around 7.4. So the buffers it uses to maintain the right CO2 atmosphere is through the bicarbonate buffer. Sometimes we add heaps if we don't have access to a proper CO2 incubator, and that is great if your cells can tolerate it, but some cells don't like it, so we can't use it in that case. For our energy source, we have glucose added to it. It will have we have this in low sugar and high sugar formulation, so you can choose the one you need for your experimentation. Now, in addition to that, we can add several other components to our basic growth medium. The essential in there is obviously the stuff that we talked about, but now we can add L-glutamine, which is an essential amino acid that the cell cannot synthesize by itself, and it is an important energy source in the citric acid cycle. It's also obviously used in protein synthesis. It is unstable in liquid media, so if your cells have a really high requirement of L-glutamine, then we will add additional L-glutamine to the media to provide it with enough L-glutamine kind of in the media so that it doesn't run out of it. We can also add non-essential amino acids when they are needed by different types of cells, especially if your cells are kind of stressed and not growing well. Sometimes providing them with these non-essential amino acids reduces the burden on the cells themselves to metabolize them all the time themselves. Finally, obviously, all cells are going to require some growth factors and hormones. There are two ways that we can provide those to the cells. One way is by adding specific growth factors and hormones associated with that particular cell line. For example, you can add insulin to it or fibroblast growth factor to the fibroblasts. So specific growth factor just for that cell line. Other times, we use a cocktail. Most times, actually, we use the cocktail, and we'll talk about that in just a second. Finally, for most cultures, especially when students are working with them in class setting, we add antibiotics and antimycotics to them. And they can be, it's usually, again, a cocktail of penicillin, streptomycin, gentamicin, and amphotericin B, and they reduce the risk of bacterial and fungal contamination. We do not like to use them all the time in culture because the cells can become antibiotic resistant and obviously start changing their phenotype, and they'll still get infected, which won't help. However, when we are doing experiments in class, I will add those to the cell culture to make sure that you have a better chance of keeping them clean and without infection. Now, for growth factors and hormones, as well as for a host of cell attachment factors, we use fetal calf bovine serum, FCS or FBS for short. This is extremely expensive, so we always want to be judicious in the amount that we use. However, it is extremely useful for growing cells in culture. It provides growth factors and hormones because it is obviously from a fetal calf, so it is full of these things. It also has cell attachment factors inside it. It also binds and neutralizes toxins, so it can be instrumental in helping your cells grow. However, since it is from an actual organism, it can contain infectious agents, and different lots are going to be different. It's not always going to be the same. It's variable in composition. So sometimes you get a lot that works better than others, and so you have to adjust the amount that you're using. It is extremely expensive. However, they do heat and activate it to make sure that there is less chance of infectious components getting taken over into your culture or for human contact. Just be aware that it is not going to be completely biohazard free, right? So it is a biohazardous material. Just like anything else, we have to use care in the amount that we use. Too much of even a good thing can be a bad thing. Usually we use anywhere from 1 to 10% of FBS in our cultures to grow them. Typically for our cell cultures, we use around 5% FBS. Next, are vessels that we grow them in. These come in various shapes and sizes, obviously. We can grow them in flasks. We can grow them in plates. The plates can be, 100 millimeter or 60 millimeters square, any different type of, sizes. They can come in these plates that have individual wells, and those come in 6-well plate or a 12-well plate or a 24-well plate or a 96-well plate even. Each one of these cases, it just gives you multiple places to grow the cells. So I could use this to have 4 different conditions and run that experiment in triplicate with N of 3 per condition, right? So this allows us to do our work in a more organized manner, and we choose the type that we need for whatever experiment that we are doing. If we need more cells to grow, we use bigger containers. If we need less cells to grow, we use smaller containers. If we need replicates, we try to use plates so that we can keep all the conditions, as many of the conditions, uniform across the board as possible. You can also grow them on slides or cover slips that are coated with ECM material also. And then for our incubator, we use a water-jacketed CO2 incubator. The inside chamber will have a water chamber in there, so to maintain humidity. It has shelves to put your plates in. It has a HEPA filter on top to maintain sterility. It has this inner glass door to close to keep that area consistent as much as possible. And then it has this outer insulated chamber. This chamber has 2 walls. Between the inner and the outer wall, you have water floating through it in tubes, and that just contains a more constant temperature throughout the incubator. That's why it's called a water-jacketed incubator. And then you maintain the temperature that you need to maintain your environment. Typically, these are set to 37 degrees and 5% CO2 to maintain an environment similar to what it is inside our body. To maintain aseptic technique, we can use laminar flow hoods, level 2 biosafety cabinets, or we can use Aseptic Space with Bunsen burner on our own benches when that is not available. Obviously, they are not going to provide the same level of, aseptic space. You have to be a lot more careful here than here, but you can still maintain aseptic technique by using that environment. In a laminar flow hood, you have a special airflow to maintain aseptic space as much as airspace as much as possible. It has a glass shield, so you only put your hands in through that little space between that gap between the hood and the glass shield. It has HEPA filters to clean the air supply constantly, and then you usually have a UV light that you switch on to decontaminate the space as well when it's not in use. Here is an example of how you would be working in that aseptic space. Obviously, you would be wearing gloves and goggles just like we always would. Now, to culture our cells, we typically have frozen stock in either liquid nitrogen or -80. We revive the cells from this frozen stock, maintain them using aseptic technique, subculture them as needed, and then freeze them down when they are no longer needed. To passage the cells, the basic reason is that we do not want to overgrow them, and we do not want them to be continuously in passage when they are no longer needed for experimentation. So to do that, we grow our cells to about 60 to 70 confluency, maximum 80% confluency. We do not want to take cells at 100% confluency because they would be outside what we call log phase of growth. So cells in culture, they will typically grow when they are low confluence. They will not grow very well because they require growth signals produced by other cells around them. Eventually, there will be enough confluency that they will grow in a log manner. And then as they become as they start to touch each other, they will get to the point where we call contact inhibition kicks in. So they slow down their growth and eventually reach plateau as they run out of space. So when they run out of space, they shouldn't be piling up on top of each other. You want to take the cells when they're in the linear log phase of growth and not when they are already slowing down or flattening out. Similarly, you don't want to take them when there are so few of them that you can't actually even reculture them. To do that, we remove the media on top of them, the spent media, after checking the confluency. We then wash them with phosphate buffet saline. This is just salt water essentially, to remove dead cells and any residual serum and media that may be still sticking to them. Then we incubate them with an enzyme called trypsin with EDTA. Trypsin is a protease, and it starts by chewing off the proteins on the outside and lifts off the cell that are attaching to the basement membrane with the help of those proteins. However, if you keep them in trypsin too long, they will continue to get chewed up and they will die. So you have to keep an eye on them, make sure they don't get over trypsinized, and as soon as about 80% of them are trypsinized, you inactivate the trypsin by adding serum back in, or media containing serum back in. The media containing serum inactivates the trypsin by binding to the cells as well as the trypsin itself. And then you can transfer them to new culture flask or freeze them, whatever it is that you need to do. The trypsin that we use is a combination of trypsin EDTA. It could be 0.05% to 0.25% depending upon the cell line that we are using. And basically what it will do is it will start by chewing off these cell-cell adhesion molecule proteins, right? So it will break apart the cell-cell adhesions and cell matrix adhesions so the cells get lifted up. And that's what we would like them to do. Again, this is showing you the lag period of growth when the cells are not quite yet growing well. And then you have the log phase of growth, and then they become more the exponential phase, and then they slow down and stabilize when there is no more space to grow. If you do not provide them with a new place to grow at this time, if you don't subculture them, they will become unstable and they will have a rapid decline, and they would all lift off and die because they cannot keep in that stage for very long. Finally, to look for contamination, obviously we have to use strategically the antibiotics and the cells as needed only. Don't keep them all the time. And then monitor the cells all the time. Every day you should be looking at them, making sure they're growing well, The media doesn't need to be changed. The incubator is working properly and getting cleaned routinely. And then obviously make sure there's no contamination in the culture. Catch it early so they can, if there is no cross contamination to other cultures in your space also. So the chemical contamination would be media through incubator, serum, or water, and biological contamination would be from bacteria or yeast, viruses, mycoplasma, or cross-contamination by other culture. These are some examples of contaminated cell cultures. So you can have little bacteria usually literally shows up as little tiny specks that are moving around in your cell culture. The cells won't look healthy, obviously. They will be dying or they will be looking sickly. They may not be growing well. You can get fungus or mold, especially here in Florida, and that will show up just like that. You'll see their little fungal balls. You can have cell debris happening or a lot of cell death happening if your cells are infected with mycoplasma, and they show up as little colonies or balls of these bacterial cultures. And then yeast, you can usually tell because usually it's budding yeast and they'll make these distinct kind of, either in doublets or these little rows stuck together, so you can tell that there is yeast infection in your cells. So hope this helps clarify some of those issues associated with basics of cell culture and the information present in the module itself.